TCA cycle reactions. And previously what we've looked at is that metabolism is a careful balance between exergonic and endergonic reactions. And we know this idea that we have two types of metabolism, and that is catabolism, which generate energy, and anabolism, which require energy. So things like palmitate biosynthesis would be anabolic because we're building up biosynthetically, we're building up a material, whereas glycolysis, TCA, those are catabolic because we are taking a material and breaking it down and releasing energy from it. And that energy is going to be captured primarily in the form of ATP, although we will be using electron carriers such as NADH in order to make an ATP, and that we can calculate reaction energy requirements as delta G. So release of free energy would also tell us how much energy such a reaction would require. So acetyl-CoA, this is a very important cellular metabolite, and it's used in the transfer of acyl, but two carbon, hence the term acetyl groups. So we're looking at two carbon groups, and we see, for example, when we take glucose and we break it down to glyceraldehyde 3-phosphate, which is a triose that has three carbons, the glyceraldehyde 3-phosphate will be converted to pyruvate, that's also three carbons, and then we're going to convert pyruvate to acetyl-CoA through decarboxylation, actually oxidative decarboxylation. So we're going to pull off a carbon, a CO2, and that will give us acetyl-CoA. So some of our primary important sources for acetyl-CoA are obviously pyruvate, but as well as the fatty acyls. So in beta oxidation, we will be breaking down fatty acyls to release acetyl-CoA at the end of those reactions. So here we go with the tricarboxylic acid cycle. So you'll know you're completed with this lecture when you can do the following. You'll need to know the sequence of reactions that define the TCA cycle, as well as the structures of substrates and products. This really isn't anywhere near as difficult as it's going to seem, because each step is a simple modification of the previous step. As well, you will want to know what types of reactions we're going to look at. We're going to look at several oxidation reactions, isomerizations, and in fact decarboxylations. Those are the principal types of reactions. We do have one synthesis reaction where we're going to combine some material, but other than that, it's basically two decarboxylations, a few oxidations, and some isomerizations. You'll also want to know that TCA does more than simply provide these electron carrying intermediates for the electron transport chain. We're going to show you what other things TCA cycle can provide, and briefly we're going to talk about regulation. TCA cycle regulation is not very well understood, but we do have some idea of what goes on, as well as the rate limiting step. So here we are. Our large central line here is showing us glycolysis, and we can see that this large cycle, typically shown in the middle of a biochemical metabolic diagram, is TCA. The other cycle over here is urea cycle, but this is our big main central point for metabolism, is the TCA cycle, as you're going to see. So this looks like a big complicated diagram, but it's really not. It's showing you a couple of things, so I'm not going to walk through it in detail yet, because we're going to go through all of these reactions in turn. But essentially what this diagram will help you do is follow the acetyl-CoA. So this molecule here, we have our 2-carbon acetyl-CoA. It's acetyl attached to our coenzyme A, and that's going to combine with oxaloacetate, which is 1, 2, 3, 4 carbons, giving us our 6-carbon citrate. Citrate can be used in fatty acid biosynthesis, as well as other destinations. But notice that we have the green part of acetyl-CoA is still being shown. So then we have isocitrate down to alpha-ketoglutarate. We lose a carbon through a decarboxylation, but notice that is not a carbon from acetyl-CoA. Those carbons are still here. So we've gone from 6 carbons to 5 carbons. Then we lose another carbon dioxide. We're down to 4 carbons, and this molecule succinyl-CoA, it's the same number of carbons as oxaloacetate. So we have oxidative decarboxylation here, oxidative decarboxylation here, and we're down to the same number of carbons as oxaloacetate. But notice the acetyl-CoA hasn't been touched. Also notice we lose our green coloring because this molecule, succinate, is symmetrical. So I can't tell you which of these two carbons were the added acetyl-CoA. So we're going to just do a series of isomerizations. Here's another oxidation, isomerization, isomerization, oxidation, and then we have oxaloacetate, and we do the whole cycle again. So we'll go over each of those steps in turn, but be aware that you can see the addition of acetyl-CoA is not the carbons that are going to be pulled off. The carbons that are going to be pulled off come from oxaloacetate, and I'm going to show you that a little bit more later on. So where do we get acetyl-CoA from? Well, acetyl-CoA can come from almost any source. Certainly fatty acids will provide it through beta oxidation. Amino acids can provide it through transamination reactions, which we're not really going to cover at this time. Pyruvate will become it through pyruvate dehydrogenase, and pyruvate's a glycolysis product. So we can see that we get the principal carbon carrier of the cell from just about any biomolecule. So we can get it from amino acids, fatty acids, as well as sugars. And through a little bit of metabolic juggling, you can get acetyl-CoA from nucleic acids as well, but not quite as directly. So essentially, all of metabolism can be geared towards the creation of energy, because as we're going to see, acetyl-CoA, which is our principal input material of the tricarboxylic acid cycle, is going to provide the electron carriers needed for the electron transport chain. This is all happening in the mitochondria with TCA, ETC both happen in the mitochondria. And those electron carriers create a electrochemical gradient, which is going to be used to generate ATP. Acetyl-CoA is very, very useful because it has a thiol ester bond, and this is a high energy bond. So just looking at the structure, we can see this is a very large molecule for a small molecule. It has a lot of charge sitting on it with these phosphate groups, a lot of polarity. This is a very large and hydrophilic molecule, so we would not expect acetyl-CoA to cross the cell membrane due to impedance from the hydrophobic core of the cell membrane. But all of this is to help create this high energy bond here. This is your thioester, and this bond is what enables the ready transfer of acetyl groups off of CoA onto just about anything else. So where do we get it? Well, as we've already seen, we're going to get it from pyruvate, which comes from glycolysis, and the enzyme is the pyruvate dehydrogenase complex. It's going to convert pyruvate to acetyl-CoA. This is an oxidative decarboxylation. So we're going to see release of CO2, and we're going to see oxidation. So we are taking acetyl-CoA from glycolysis, which is sometimes called an energy generating pathway. I object to that. It is an emergency energy generating pathway, but it's not a very good energy generating pathway. Acetyl-CoA is going to enter into the TCA cycle, which you may hear also referred to as the Krebs cycle and the citric acid cycle. So we're getting pyruvate or fatty acids to generate acetyl-CoA. Acetyl-CoA is going to go through the TCA cycle, and we're going to remove 2CO2. We're going to get 4 reducing equivalents for the electron transport chain. We get a little bit of GTP, and we do a whole lot of isomerization. Then ultimately, you're going to get about 9 or 10, depending on where you look, ATP from this process, from one turn of this process. So TCA is found in the mitochondria, although you will see some isozymes in the cytosol. One of the big ones is malate dehydrogenase, has an isozyme in the cytosol as well that is used in palmitate biosynthesis. The pyruvate dehydrogenase complex is also found in the cytosol. And we have 4 reactions that transfer electrons to either NAD+, which is most of them are FAD. These molecules, in turn, NADH and FADH2, once they're reduced, are going to feed into the ETC through either complex 1 or complex 2, and that creation of redox reactions ultimately to reduce oxygen to water is going to create a proton gradient that is used by the F1F0 ATP synthase to generate ATP. So just real quick overview of all of that. We have fatty acids, glucose, amino acids, and as I say, with a little bit of juggling, you can even get nucleic acids to do this. We'll give you acetyl-CoA. Acetyl-CoA is going to go through the citric acid cycle, as shown here. I more formally call it the tricarboxylic acid cycle. And you're going to get some CO2, electron carriers, GTP, through the electron transport chain, through which we're going to convert oxygen to water. This will create our proton gradient, and that gradient is used by the ATP synthase to add inorganic phosphate to ADP to form ATP. So our input material is acetyl-CoA. Each turn of the TCA cycle is going to result in NADH. You'll get 3 of those. You'll get 1 FADH2, you get 1 GTP, and you get 2 carbon dioxide. Your big key idea is that this cycle really is generating NADH, and it's driven by the creation of NADH. NAD can be rate limiting if we start to run low, so you can see there is an intimate connection between the electron transport chain and the TCA cycle, and we'll show you that in a moment. So here we go, the TCA cycle. After all that preamble, the TCA cycle. So our first idea is how the carbons are being used. So we're starting with oxaloacetate as the final product of the TCA cycle, and it's going to combine with an acetyl group, acetyl-CoA, to form citrate. So we go from 4 carbons plus 2 carbons to 6, split off 1 CO2, that's 5, split off another CO2, that's 4, then we do a little bit of oxidation, isomerization, and you're back to oxaloacetate. These carbons that came off did not come from acetyl-CoA. They come from the previous oxaloacetate, as I will show you. Our metabolites, pretty straightforward. This will help with understanding the enzyme names. Citrate goes to isocitrate, goes to alpha-ketoglutarate, goes to succinyl-CoA, goes to succinate, goes to fumarate, goes to malate, goes to oxaloacetate. Oxaloacetate combines with acetyl-CoA to give you citrate, and so on. So these few images I'm providing are to help you kind of get a large-scale sense of what's going on in the TCA cycle before we get down to the nitty-gritty. And so here we are at the nitty-gritty. So these are the reactions and structures. So let's take a look. So here's acetyl-CoA, and we have oxaloacetate. Whoops. Sorry about that. Acetyl-CoA and oxaloacetate. These combine through citrate synthase to give you citrate. Now notice these are the carbons from the acetyl-CoA, and I'm going to go ahead and highlight these carbons throughout the rest of these reactions so that you can see the CO2 does not come from the acetyl-CoA. So we have acetyl-CoA combines with oxaloacetate, we get a citral-CoA intermediate, we're going to add a little water, so this is a hydration, and we get citrate. Okay, so you can see we lose that double bond. There's our hydration. So we get citrate, and we release CoA in the process. So some of the energy from this is going to come from the CoA thioester bond. So now we have citrate, and boxed in here is our acetyl-CoA. We're going to use the enzyme aconitase. We're going to remove a water to give an aconitate intermediate. Notice the appearance of the double bond here. Okay, and then we're going to add water back in. So we've pulled off this water molecule here to create the double bond. Then we add water back in, which allowed us to move the hydroxyl group. So the hydroxyl group moves from this position, the third position, to the second position. Now notice, why is this called the tricarboxylic acid cycle? Because we have 1, 2, 3 carboxylates. So tricarboxylic. So in this step we have converted citrate to isocitrate. Notice it's an isomerization. We split out of water, we rehydrate in order to move the hydroxyl group. The way this is maintained is that actually intramitochondrial amounts of oxaloacetate are kept low, and the citrate synthase reaction is the delta G of this reaction is really low. It's very negative, highly exergonic. So citrate synthase, this reaction here, is actually pulling the entire process forward because we have low amounts of oxaloacetate. So you might think that this would tend to pull back to oxaloacetate, and it does. However, for a couple of reasons. One, that the delta G of this reaction is extremely negative, and part of that is coming from the thioester, and that citrate itself is used very readily, that this entire cycle is pulled forward by this reaction. So all of TCA is being pulled forward by the activity of citrate synthase. So citrate to isocitrate actually favors citrate. So let's go back and take another look. So we have this sort of free-flowing reaction, and this actually favors citrate. So the formation of citrate is actually favored from both directions. That should give you a clue as to how important citrate actually is. Citrate can be exported from the mitochondria. One destination is going to be palmitate biosynthesis in that case, but we can also convert it back to oxaloacetate in the cytosol. So citrate has a lot of metabolic destinations. It's a very important intermediate. So aconitase is going to create isocitrate, but it's going to favor citrate. So here we are at isocitrate. Notice I've still got that green box around the acetyl-CoA. And here is going to be our first decarboxylation. And notice this carboxylate is not coming from the acetyl-CoA. So we have isocitrate dehydrogenase that is going to do an oxidation. And we do this down here. And that gives us our double bond that we see here. We're going to release proton and NADH, as well as, in our second step, we're going to release the CO2. So we have our first NADH and our first CO2, and our product is alpha ketoglutarate. Alpha ketoglutarate, notice I've still got your acetyl-CoA marked off, is going to combine with coenzyme A using the enzyme, the alpha ketoglutarate dehydrogenase complex. Notice it's a dehydrogenase. When we see dehydrogenases, we have one here, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, we should be looking for an electron carrier. And indeed, we have one. So we're going to do another little bit of oxidation here, as well as decarboxylation. So we lose another CO2, and we get our restoration of a thioester bond as we move this material onto CoA, giving us succinyl-CoA. So isocitrate, isocitrate dehydrogenase, which also decarboxylates, gives us alpha ketoglutarate. Alpha ketoglutarate goes through alpha ketoglutarate dehydrogenase complex, which gives us another oxidation and another decarboxylation to give us succinyl-CoA. Now, this reaction of isocitrate to alpha ketoglutarate, this is the isocitrate dehydrogenase reaction and strongly favors alpha ketoglutarate. This actually is the rate limiting step of TCA. You might be inclined to think it's citrate synthase, but it's not. The formation of citrate is just highly favored, extremely highly favored, and helps pull the entire cycle forward. But the rate limiting step is isocitrate dehydrogenase. As we've seen before, this is going to require NAD, so levels of NAD are going to have an impact on the rate of this reaction. We're going to generate some CO2, and we're going to generate NADH. And just as a side note, this enzyme does require manganese or magnesium for the reaction. Now going onward to succinyl-CoA, alpha ketoglutarate to succinyl-CoA is very highly favored and is performed by the alpha ketoglutarate dehydrogenase complex. From here we get our second molecule of CO2, so that's another decarboxylation, and a second NADH. So what we have, just going back, are two very strong forward reactions that help pull things away from citrate. Because remember, citrate's kind of a sink. Reactions that form citrate are highly favored, and so we need to be able to pull away from citrate. Yes, citrate formation is pulling the entire cycle, but remember, citrate to isocitrate favors citrate, which could actually impede the cycle somewhat. So these two very strong forward reactions pull materials away from citrate, and then the formation of citrate pulls these materials forward even further. Remember, this is a cycle. So now we're at succinyl-CoA. I still have our acetyl-CoA marked. and we have succinyl-CoA. So succinyl-CoA synthetase, now this is a bit of an odd named enzyme because it's more or less being named for the backwards reaction. But that's okay, we've seen this happen before. So we're going to take some water, inorganic phosphate, and convert GDP to GTP, and you get succinate. Now, while I've marked the acetyl-CoA, you would do well to sort of recognize that this molecule is symmetrical. So at this point, if this molecule is released, which it will be, you can no longer determine which carbons originally came from the acetyl-CoA that entered this series of reactions. I'm showing them there just so you remember there is still an acetyl-CoA carbons present, but we can no longer tell which two carbons are the acetyl-CoA carbons. Also note we are now down to 4 carbons. That's the same number of carbons as oxaloacetate. Actually, we were at 4 previously, but I'm going to draw your attention to it now because we're about to do the isomerizations. So the CoA is no longer involved, and we have succinate. Again, this is not saying that these carbons are the acetyl-CoA carbons. Just be aware we have acetyl-CoA carbons still in the system. So we're going to use succinate dehydrogenase now. And since it's a dehydrogenase, we should expect an electron carrier. Indeed, we do see one, FAD to FADH2. Now, in the electron transport chain, FADH2 enters via complex 2. But complex 2 is succinate dehydrogenase. So succinate dehydrogenase will also be using FADH2 to restore FAD, and that thereby reducing ubiquinone to ubiquinol. So succinate dehydrogenase, this is complex 2, and it can also be called the FADH ubiquinone oxidoreductase, but usually when it's being referred to by that name, the focus is on the electron transport chain. If it's being called succinate dehydrogenase, the focus is on TCA. Context matters, but be aware, it is the same enzyme. So by doing this oxidation, you can see we're pulling off these hydrogens, we get, and one will come off as a proton, we get our double bond. Again, we have an achiral and fully symmetrical system. So succinyl-CoA is converted to succinate by succinyl-CoA synthetase, or sometimes called succinate thiokinase, or sometimes called the FADH ubiquinone oxidoreductase. We actually do make some GTP in this reaction. That GTP is not an ATP, it is a GTP, though it may be considered an ATP equivalent by some sources. This GTP, for example, could be used in the mitochondria for protein synthesis. Translation of mRNA requires GTP, so here's a source of GTP. The reaction is freely reversible, as you can imagine, even though this arrow is showing that it isn't, but it is, and we do have energy sources in the form of GTP. Now we're on to our oxidation reduction event. So succinate is going to be oxidized to fumarate by succinate dehydrogenase. This uses an iron sulfur cluster. Strongly inhibited by the presence of malonate, which is going to be a competitive inhibitor here, and oxaloacetate, which will slow down these sets of reactions. It can be, interestingly enough, activated by ATP, the presence of ATP, which seems counterintuitive, as well as inorganic phosphate and succinate. Succinate's not entirely surprising. We overload substrate where we should expect the reaction to push forward. It is interesting that ATP accelerates this reaction, though. A little counterintuitive. So now we're at fumarate, and we're going to use the enzyme fumarase. Couldn't ask for a better named enzyme. We're going to add some water, so this is a hydration reaction, and we get malate. You can see now we have a chiral carbon, so this is L-malate. We have malate. Again, this box is not indicating these are specifically the acetyl carbons. I'm just showing you the acetyl carbons are still in the molecule. So malate will then go to malate dehydrogenase. Since it's dehydrogenase, we should expect an electron carrier. Indeed, we see NAD again. And now we have oxaloacetate again. So 2 of those carbons came from the acetyl-CoA that entered the reaction. The 2 carbons were from the previous oxaloacetate, so the original oxaloacetate that was used lost 2 carbons, then recombined with acetyl-CoA to make oxaloacetate again. So what I want to show you, I'm just going to revisit, I'm just going to sort of continue the cycle. So let's consider, I've now highlighted in red, let's just say the acetyl-CoA from the previous cycle, which is now located in oxaloacetate, that oxaloacetate is going to combine with acetyl-CoA through citrate synthase to give us citrate. Notice here is our new acetyl-CoA, and just for the sake of argument, let's say this is our old acetyl-CoA. We don't actually know, but let's just say that it is. So then, when we go from citrate to isocitrate, here's our acetyl-CoA, here is our original acetyl-CoA, and what I want to show you is that there is a carbon coming off possibly of the acetyl-CoA from the previous cycle. So in order to start seeing carbons come off of acetyl-CoA from the previous cycle, it takes at least, at a minimum, 1 round of TCA before a carbon from that acetyl-CoA comes off. And you'll notice we get 1 carbon off, and then we have alpha ketoglutarate, we go, and there it is. We're now back to kind of what we were expecting. So to get both carbons degraded or decarboxylated from an inputted acetyl-CoA will take, at a minimum, 3 cycles, as I'm going to show you here. This is a minimum. So how many rounds of TCA for each carbon of acetyl-CoA to be removed? On the first round, when we add the acetyl-CoA, you're not going to see any carbons from that acetyl-CoA removed. On the second carbon, you may see one of them removed. On the third round, you may see the second one removed. Now, that could be a little bit different because we know we have that symmetrical molecule. It's really hard to say, but these are minimums. These are when we would expect things to happen. So in other words, for oxaloacetate, carbons are removed, then acetyl-CoA replaces those carbons. So we do have a very strong endergonic reaction in this system. So L-malate to oxaloacetate is mediated by malate dehydrogenase. We know that. But while we do get NADH, which is going to be consumed, this reaction actually favors malate. So it's one of the reasons citrate synthase pulls the system forward. If citrate synthase was not as strong as it is, then the endergonics of this reaction could actually pull the TCA cycle backwards. So if citrate synthase isn't working for some reason, we might expect to see elevation of L-malate, which is a bit strange because if you look at the cycle of reactants, you would not expect this to happen. You'd expect, well, if citrate synthase isn't working, if we're not making citrate, we might see oxaloacetate buildup. However, oxaloacetate from malate is not favored, which means buildups of oxaloacetate will backflow to malate. Okay, so after that quick quiz, hopefully you did well. If not, please go back and review those questions. Help getting you started in thinking about how those reactions work and how they are controlled. So now, kind of energy are we going to get out of this? So you'll probably see the number 10 kind of bandied around. You get ATP being 10 ATP being formed between NADH, Fe2, and GTP. Now I maintain GTP is not ATP, but it can be considered a GTP to ATP equivalence, which does happen. So it's not entirely unreasonable, but do remember the TCA cycle does not literally create ATP, it literally creates GTP, which can be converted to ATP. However, TCA makes a lot more than just reducing equivalence and some energy. It is a central cycle of metabolism, and it makes all kinds of precursors and other materials. Just to give you an example, oxaloacetate can go on to amino acids, alpha ketoglutarate can go to amino acids, specifically glutamate, and then on to neurotransmitters. Succinyl-CoA goes on to make heme. Malate can be used in gluconeogenesis. Citrate goes on to fatty acids and sterols. And there, as you can see, we have all kinds of outputs from the TCA. This is why TCA reactions are so important to survival of a cell. We can't really have failures in the TCA. There aren't many diseases, and the few that do exist are extremely dangerous. So just to briefly revisit this, we get succinyl-CoA that can be made into heme. Malate and oxaloacetate. Oxaloacetate is going to be transaminated to aspartate, and that can give you, aspartate, which can give you asparagine, which can then go on to uracil, cytosine, and thymine. Oxaloacetate can also become phosphoenol pyruvate and used in gluconeogenesis to make sugar. Alpha ketoglutarate will become glutamate and thereby can become glutamine and in fact other amino acids as well. Citrate, as we've seen, can go on to be converted into palmitate via acetyl-CoA. So how is this cycle regulated? It's still a bit cryptic. We do know the presence of acetyl units is a key factor in determining rate, obviously. We need to be able to combine acetyl with oxaloacetate to get citrate. We know the reaction of citrate is very, very strong and pulls the cycle forward. We know we need to pull electrons off of NADH and FADH2 since we need those and their oxidized forms for the dehydrogenases. And we know that respiration is going to be very highly controlled by availability of ADP and inorganic phosphate as well as oxygen. So if we're lacking ADP, we can't pull this cycle forward. If we're lacking inorganic phosphate, we can't pull this cycle forward. If we're lacking oxygen to convert to water, we're going to see backups, and then we're going to see buildups of NADH and FADH2, which will slow the cycle down. So the flow of electrons in the electron transport chain is going to play a very big role in TCA cycle regulation, TCA cycle activity. However, that's coarse control. That's just large-scale control. We still don't quite know the fine-tuning, and that creates some interesting problems. As we saw, and it's not shown in this figure, that actually ATP can accelerate parts of this cycle, which is unusual, but maybe it ties into biosynthetic reactions and not energy production. We can see that NADH and acetyl-CoA can also inhibit the creation of pyruvate. We see succinyl-CoA can actually act as inhibitors for citrate synthase. We see energy control over here with ATP being a negative regulator and ADP being a positive regulator. And calcium is going to play a role. Elevated calcium. Where's that coming from? Maybe from sarcomere contraction, release of calcium from the sarcomere reticulum. It's really unclear. It just does seem to play an accelerating role. We see some feedback inhibition. That's not terribly surprising, but all this shows is that control of TCA is very complex. We do understand some basics, such as NADH and FADH2 and presence of acetyl groups, and of course just respiration overall is going to play a major role. The rate-limiting step is the NAD requiring isocitrate dehydrogenase, which is going to be stimulated by the presence of low energy signals such as ADP or AMP, and be inhibited by the presence of energy within the mitochondria, ATP or NADH. So in summary, just to kind of bring this all down, The central metabolic cycle is TCA, and we're going to fully oxidize the two carbon moieties of acetyl-CoA to CO2, but you're not going to do that the round the acetyl-CoA is added to oxaloacetate. That's going to take time. We do see that about the equivalent of 10 total ATP is going to be formed, although bear in mind one of those ATP is really an ATP equivalent and is GTP. And this is the type of nuance I'm talking about when I say be able to explain the story. TCA provides many metabolic intermediates that come from oxaloacetate, malate, citrate, alpha-ketoglutarate. And notice I've listed citrate twice. That's for a reason. Citrate is really important. And the TCA, we didn't cover this, but just to know, the TCA can also take substances from many different sources, not just acetyl-CoA. And any of these intermediates can also backflow into TCA as well. And it is a very tightly regulated cycle, and its key rate-limiting enzyme is considered to be isocitrate dehydrogenase. But do bear in mind, citrate synthase, being highly exergonic, does pull the entire sequence of reactions forward. And that's pretty much it for our summary. Please be sure to go back and look at those chemical reactions, look at those structures, look at your substrate names, your enzyme names, your product names. It will really help you in understanding the specific chemical reactions that are happening. Practice it, talk about it with your colleagues, work on it. Of course, please come to office hours and discuss it with me. Good luck on your studying, and keep at it.